magnet is being turned on and off with different periods. And during the period when we turn the magnet on, we can see a decrease in fluorescence of about 50%. And so we can see, basically, I can have an external magnet, let's say an electromagnet, which I'm then pulsing on and off, in this case, every 10 seconds. And that makes, in some respects, my fluorescent signal flash. And that in itself is already quite an interesting biotechnological tool. So let's look at one example. So something we've probably seen and we've heard about several times already in this conference is all kinds of applications of lock-in measurements, which broadly speaking, if we've not come across this, is where I modulate a signal of interest at a precise frequency, in this case, I suppose 0.1 hertz, and then, I guess 0.05 hertz, sorry. And then I try and extract back the signal of interest by looking in that frequency band and seeing how much of it, how much amplitude, for example, is in my Fourier transform in that frequency band. And so imagine that I have one of these cases where I have a really messy tissue or sample. There's all kinds of fluorescent things in that sample, and maybe I care about one of the green things. I don't care about the rest of it, nor do I care about red or blue. I just care about the green channel and some specific people in there. And currently, it would be difficult for me to see what's going on. But imagine I start turning my magnet off and on. I can maybe identify that that's the one I care about. And so now a messy image, which I could never really say what is what, I can pull out, actually, maybe that guy is the one that I'd like to locate in this sample. And so that's a very basic application of this kind of technology. Other things might be, imagine that I can make different proteins where, let's say, the characteristic timescale of this is different. So I could make a very fast one and quite a slow one. Well, then I could just generically expand the number of channels I can use for fluorescence microscopy, because I could have, commonly in fluorescence microscopy, I might get up to 3 or 4 color channels, like a green, a blue, red, and yellow. I can't put them arbitrarily close together because they have fairly wide excitation and emission spectrums. But if I could have within each of those channels different reporters with different timescales, I could then separate them out and basically multiplicatively expand the number of things I could measure at once in one experiment. And these are really comparatively simple applications just using this magnetic field effect. And so with that, I will pass to Gabby, who will tell us about some more complex and exciting applications.
Okay, thank you. Yeah, so once we saw this magnetic field effect, the question is, like, where is that coming from? And I guess people here have already been thinking about this a fair bit, but I'll go through it again. So in this case, we're talking about the radical pair mechanism. So it's a little bit different from the triplet sort of situation that Peter was talking about yesterday, but in the end, the result is kind of similar. So to begin with, we just have a straight fluorescent protein where you put in blue light and green light comes out. And it has this sort of triplet mixing, which can go on off to the side. And this is the radical pair mechanism where there's a flavin bound inside the protein. And we think one of the amino acids on the backbone of the protein can participate with that flavin to create this kind of singlet triplet system. And with no kind of, in just the natural state, there can be mixing between these two states. And that's through hyperfine couplings. And so in some sense, there's multiple pathways back to the ground state other than the fluorescence. However, when you bring a magnet near this protein and you create a magnetic field, that causes this Zeeman splitting, and that prevents this mixing from happening because it's no longer kind of energetically possible to go from these triplet states to the singlet state. And because the singlet state actually kind of is a faster decay back to the ground state, which you can think of as just the ground state is also a singlet state, this means that we see a reduction in the fluorescence. So this kind of explains the magnetic field effect that was on the previous slide. And so this is the hypothesis. And a good way to test this is if you can kind of turn this effect off with the magnetic field, can you then turn the mixing back on? And indeed you can. So if you apply a radio field at the right resonant frequency with this magnetic field to drive the transitions that have now been Zeeman split, you can enable these pathways again, and so the fluorescence goes back up. And so you get kind of the opposite effect if you like turn the radio on and off. When the radio comes on, you're sort of undoing the cancellation of the mixing by the magnetic field, and so the fluorescence comes back up, so you have an increase in fluorescence. And so this means we have another way to kind of modulate the fluorescence, this time using both the magnetic field and the radio field. And crucially, it only occurs when the magnetic field and the radio field are kind of on resonance. So there's this particular condition that has to be met. And this, I mean, is very standard. This is like how MRI works. And so based on that, there's a number of applications, again, kind of immediately possible. But first, just to show you how we sort of demonstrated this ODMR, optically detected magnetic resonance. So this is doing it on a wide field microscope. It's at room temperature. The antenna is a very simple kind of strip line antenna. You would have seen very similar in like an NV lab. And we get very kind of remarkable contrast just with this very simple experiment. And even at kind of a single cell resolution, you get quite a good signal contrast. So this is just blue light coming in and then measuring green light. The black dots here are from integrating over a single cell. So the contrast is quite good. This is a single sweep that took 50 seconds to acquire. So it's not like I've done tons of averaging to try and make the data look nicer. It's quite bright and there's quite a lot of contrast. So it works quite well, and the LED intensity is sort of standard for what we would use for any other kind of fluorescent imaging experiment on this. So we're not pumping in tons of power either. And because it's bright and there's a good contrast, you get a decent sensitivity. The reason that the sensitivity is like a lot lower than something like a nitrogen vacancy is the width is quite a bit wider, and that's got to do with the couplings inside FMN. Nevertheless, this opens up a bunch of interesting applications. So one is doing this kind of imaging that Harrison talked about. So can we locate fluorescent proteins in space? And the reason that you'd want to do that is, although fluorescence, optically you can look at an image and potentially see where something is, often there's other things going on. And also in the case of like trying to image in tissue, you have this problem of depth and like 3D localization. And there are ways to try and reconstruct from some fluorescent light, like where the light is coming from in 3D in a tissue, but it's very complicated. It usually requires some like a priori knowledge of the optical properties of the tissue. So it's a little fraught. What we would imagine is that you could do some kind of test where compared to a standard fluorescence label, you can now have some way to localize. And so I'll just walk through how that would work. So as I mentioned, you have to be on resonance for this to occur. So that means that the radio frequency and the magnetic field strength at the spot have to kind of match up through this gyromagnetic ratio. And so here we're moving the permanent magnet at different positions to the sample, and that shifts the resonant frequency, and you see the peak occurs at different points. Right, so, and then you could imagine doing lock-in on top of this, where you turn the radio on and off. And so if we have our happy koala here, you can imagine scanning that koala, and ideally, you can now localize basically the fluorescence of your thing to so far like a 0.6 millimeter kind of slice. You probably wouldn't do this in a koala because it's furry and not so transparent, but you get the idea. And if you were to do this with a traditional method like of fluorescence, I mean, certainly the most naive way to do it just with fluorescence, you don't get any of this special information. As I mentioned, there are some ways to try and do better than this, but it's difficult, and it's certainly not, you can't, it's very hard to just impose a gradient in free space in any other way than a magnetic field. Kind of any other gradient you can think of, requires some interaction with the material. Okay, so we wanted to test this, and we did this by acquiring an MRI coil, which happened to be lying around. So this is a small animal MRI coil, and it can create a radio field in a kind of 40 centimeter cube roughly volume, sort of at the end on the right. And this is just a long thing because it normally goes inside a superconducting magnet to provide the 7 plus Tesla fields that you would do in a normal MRI experiment. And then we also have a Helmholtz coil around the kind of area where the sample goes, and then that can create a small field of a few millitesla. So our mouse sample, not a koala sample, I guess, is this block of PDMS, and inside that PDMS, we can insert cells, and those cells can be positioned at different points along kind of the axis of this tube. So we're just going to try and demonstrate 1D imaging in this case, but you could set up gradient fields in any direction and therefore localize in any direction as well. And so we have this permanent magnet at the end, which creates a 1 millitesla per millimeter field. And again, this is a very simple setup. You could probably achieve larger gradients and therefore better spatial resolution if you wanted to. And we have some kind of custom optics to try and get this to work, because typically people don't do MRI using fluorescence. So we have to have a way of getting the light in there, which is this LED coupled to optical fibers to sort of take the light down the tube. And then this long quartz rod, which collects as much light as possible and takes it back to a photodiode. And I guess just to note that the photodiode is literally a photodiode, it's 1 pixel. So any spatial information we're getting is going to have to come from this resonance. It's not going to come from seeing an image. Right, and so this is the full assembly with all the radio parts and the electronics. And schematically, it looks like this. So again, we have kind of just the area now of the MRI coil that has the RF field. There's this gradient field created by the permanent magnet. We can shift the gradient field along using the Helmholtz coil, and the antenna's on resonance at 500 megahertz. So as you shift the gradient, you're kind of shifting where the resonance condition occurs. And so we put pieces of PDMS in it separately with the maglev cells positioned at different pieces at different points along that axis. And the localization was quite good. So you could see from the data, like when the PDMS is, when the maglev cells are on one side, we get a peak at that position in space. And then when they're on the other side, we get the other position. So you can imagine if this instead was, you know, some mice or something, some tissue, and we're trying to figure out where exactly in space is the cancer that we've labeled with our protein. 1, you get lock-in detection. 2, you get spatial information along one axis. You could then get the other 2 axes either by actually using a camera here rather than a photodiode, or just by setting up gradients, magnetic field gradients in the other direction and doing the lock-in and looking at the resonance condition that way. And also when you put the cells in at multiple positions, you can also just resolve kind of two points at the same time, although it is a bit more noisy, and that's probably just because, you know, you now have this tube of cells and this other tube of fluorescent cells, and you're looking along that way, so they're kind of obscuring each other. I think this could all be much improved with like more engineering, but the science is certainly there. Okay, so that's spatial imaging. Another application that is exciting is, can maglev sense the surrounding environment? So this has been the idea with, you know, NVs and other sensors for a long time, is you could potentially use it to measure things like reactive oxygen species in cells, look at DNA damage and this sort of thing. So we just wanted to ask the basic question, like, this protein has a flavin bound inside it. Is it at all responsive to what's going on the outside? And the answer is yes. So we mixed it with gadolinium, which is gadolinium chelated. So gadolinium is highly toxic. If you just add gadolinium, which is the first thing I tried, it just causes the protein to like unfold and aggregate and go everywhere. Gadobutrol is an MRI contrast agent, which is routinely injected into people, so it's safe. But it still has this property that it can cause the relaxation. So what we showed here is that as you increase the concentration of the gadobutrol, it reduces the magnetic field effect size, and in a sort of dose-dependent way, which is what we would expect with spin relaxation. The concentrations are, of course, quite high. This was with purified protein, and as I say, it's really just a test that shows, yes, maglev is responsive to the surrounding environment. Much work to do to get it to the point of actually sensing something interesting inside the cell, but I think this is a nice first step. And so finally, we've done a bunch of kind of spectroscopy and characterization of maglev. I don't want to speak about it too much here because also I'm not the expert in this. Our co-author, Anna, is. So if you kind of scan this QR code or look at the paper when it comes out, there's heaps more detail. But for example, one thing that we've looked at is like the photostability of maglev. So this is quite interesting for people who have been studying cryptochrome for a long time. So this protein for magnetoreception in birds, it's not very stable. The flavin tends to kind of go into a photobleach state very quickly. Whereas here, we can image for a very long time. So I mean, this is 2 minutes here. And over here, we have like 80 minutes to 180 minutes showing the kind of pathway of the FMN as it goes from sort of this usable FMN to the semi-Queenan state and then to FMNH minus, where at that point you can't do spin radical pair stuff anymore. And it's just interesting to see that this happens over quite a long time scale. So photostability-wise, maglev is quite stable. Yeah, thanks very much.
All right, so just in the last 5 minutes to try and kind of sum up and point a bit more to the other general research we have going on in Oxford. So our lab is kind of half made of people doing synthetic biology, so genetic engineering, mostly in bacteria, yeast, protein engineering type things. But then separate to this, as you may have seen, we're doing various instrument building, computational and kind of AI work, and then different actuation hardware. So building robotics to try and automate some of this stuff. So for example, our a bit ridiculous RGB microscope, which is the one that's been used in all of the data, Gabby just presented to other machines like evolution machines. We've recently overtaken the Lenski experiment as the longest ever evolution experiment with some collaborators at Harvard who've been running one of these machines that we designed for evolving bacteria. Maybe you don't care about the evolution, but in terms of the directed evolution, for making better and different versions of these kind of fluorescent proteins we're talking about, there's lots of different approaches people have taken. So the one used to make maglev by Maria and co is kind of like put things on a plate, put a bunch of variants, and then try and pick out the one that you like the most. You could imagine doing that in test tubes, but it will be incredibly slow and quite painful. You can't really do that in the workhorse technique. So if I'm trying to change my fluorescent protein brightness, for example, in this field, mostly people would use fluorescent cell sorting, which is not really going to cut it for this kind of work because you get one look at a cell very briefly, then you lose the cell. So you're not going to be able to measure a dynamic phenotype on the order of seconds. You need to have lots of measurements to do this, which is why you need a sample that's kind of fixed somehow. And so at least in our opinion, these are reasonable methods, but they have a lot of flaws. And so kind of separately to this specific maglev work, our team has spent a lot of time developing microfluidic screening systems, which are basically like a very small microfluidic chip with a whole lot of channels, each which contains 5 to 8 bacterium with the top one trapped. And therefore this bacterium you can watch for several days at a time, taking maybe 100 images a second if you really wanted to probe one very quickly. And so here's a long time lapse of several hours. You can see the cells growing and being moved out by a flow past them. But the beauty of this technique is you can run like 100,000 versions at once in a chip, and you can image 100,000 versions at once, and do that quite regularly to measure this kind of phenotype. So for example, imagine I have this fitness that I care about, like how big is my contrast? I can measure this. I can use my computer dynamically during the experiment to pick which ones I like. In our system, you can then use like a UV LED with a micro mirror to go and kill all the ones you don't like. So you can impose selection upon the whole population to get the ones back that are good. You can then propagate them and make them mutate some more and then go around this circuit. And so this is a kind of method we're developing further to apply to these fluorescent proteins, because you can then in theory do many hundreds of thousands at once and really push down very hard on phenotypes that you care about. This is kind of an evolution one. You can also just run this as a screen. So another technique being developed is make a big library of cells, do a similar measurement in the same chip, but then do on-chip sequencing to get back all of the function to sequence relationships. So here you can imagine doing 100,000 or more variants in one experiment, looking at them for 24 hours with all kinds of different chemical, magnetic, RF environments, and then getting back the sequences for all those proteins, which we believe will be very powerful for understanding mechanisms of these systems in the future. So finally, in terms of future development, I suppose we're very interested in collaborating with people that are interested in these kind of effects. So, we've built these systems which are really suitable for engineering dynamic effects in proteins and bacteria. So anything from 100 hertz timescale to like many hours or days long, because you can make these nice measurements where cells are trapped and you can see what they're up to even quite quickly, like in the maglev case. And also if you're interested in other kinds of systems which have these kind of phenotypes, whether or not they're fluorescent proteins, we're keen to hear. And similarly, if you're interested in developing further some of these tools, we're very happy to share protein sequences or actual samples of plasmids and cells, also help with hardware development if you guys are trying to implement this around existing tools in your lab. So with that, I think I'm at time, so I better say thank you, so especially to Gabby, who's led a lot of the work in this project, but also our broader team of postdocs and PhD students in Oxford and collaborators who've helped a lot throughout this project. So thanks very much, everyone. I think I'm also the chair, so any questions?
Thank you both for the beautiful talk. Is it possible to integrate this system with maybe small animal MRI to leverage the complementary contrasts? And also you can leverage their hardware.
I mean, that coil that we had is a small animal MRI coil.
I see.
Designed for like a mouse model. We don't need very big gradients. So like the gradient here is a millitesla per millimeter or something. So compared to MRI, it's not really necessary. It would give you better spatial localization, but then it may make other things more difficult, like the sensing and so on. Mm-hmm. So this is something we're going to pursue and looking at how we can actually make this work on a real mouse, not just a kind of fake puck mouse. But yeah, down the line, it will be very interesting to do that and also look at optimizing the signal to noise, for example, making better contrast, more bright reporters, stronger ODMR contrast. Currently, these have all been engineered for the magnetic field contrast, the ODMR contrast. Mm-hmm. Getting faster response times, so you can do more lock-ins more quickly, which will also improve your signal-to-noise ratio. Mm-hmm. So there's a lot of biological engineering and also instrument engineering to do to make that a useful technology in an actual living thing.
Right. Are there any intrinsic proteins that have this type of properties? If so, can we find one that has a shorter response time? I think yours is on the second scale. I suppose
like the cryptochrome and stuff people have looked at for this. As Gabby said, it has problems with bleaching. It doesn't last very long. This one is very good compared to any other protein that we know about in terms of these metrics that we care about. And actually, if you look in the paper, there is proof that you can improve the response time with engineering. And so this has not been pursued a lot. Maria did one screen for it, I think. So pushing down on this more could then make it much faster, and then you get better measurements in general.
Great, thank you.
Yep.
Yeah, super cool. Thank you. You highlighted that the line width for when you apply the microwave pulse is quite broad, and so it's inhomogeneously broadened. Is that width coming from just inhomogeneities in the magnetic field, or are there molecule to molecule differences for where the maximum in that microwave frequency is?
Yeah, so there's a few things, right? One is, of course, it's not a crystal, so you have just tumbling. I think the majority is probably just from the couplings in FMN, so okay. It's not like an interaction. But honestly, we don't really know much because like all we've done is kind of got this ODMR, yeah. And there's very little that's been done beyond that so far.
Have you looked for correlations between the microwave transition and the emission peak? That might be. You probably have the data to do that already. That might be a cool experiment. Yeah.
Last question, so we can move on.
Oh, congratulations on the significant results. I wanted your insight on the voltage imaging community, which is just the optical voltage imaging community from about 2005. Multiphoton microscopy is at a level where we can image 1 mm deep inside the cortex, voltage imaging at scales of milliseconds, and diffraction limited in moving mouse animals. So I wanted your insight on how this technology is going to scale, because we lose a lot of these important photons in contrast to optical imaging. About 90% of the photons would be lost. And as long as the excitation and the readout is optical for qubit proteins, the flavin and also the EGFP counterparts that we were discussing yesterday. So in what situation do you see this this technology eventually?
general, you're right. At some point,
Thank you.